very good morning to one and all present here. I am Sandra Chelsea Saji of class 12B. I'll be teaching biology today. My topic is gametogenesis. So, can you name the process by which living organisms make their own copies, of the same individual? Reproduction. There are two types of reproduction. Can you name those? Yeah, sexual and asexual reproduction. In sexual reproduction, we need two things. What are they? Yeah, a male gamete and a female gamete. Together they fuse, and that results in the formation of a zygote, and that is sexual reproduction. So here I'll be telling you how the male gamete forms in the body of the male. So gametogenesis, it is basically the formation. The word genesis means formation. So gametogenesis is the formation of gametes, and there are two types of gametogenesis, as I told. One is spermatogenesis and the other one is oogenesis. Spermatogenesis, as the name suggests, it is a formation of sperms, and oogenesis is the formation of ova. So I'll be taking spermatogenesis today, only spermatogenesis. So, the unit of reproduction in the males is the testes, as you know. They have a pair of testes, that is, two testes are there. Like you can see here in the picture, the section of the longitudinal section of the testes. So, two testes are there. Right now we'll speak about one of them so that you will be able to understand. So, one testes, you're taking it, okay? There are many compartments, approximately about 250 compartments are there in one testis. This is known as the testicular lobules. Okay, so one compartment is known as the testicular lobule, and inside the testicular lobules, inside one testicular lobule, there is approximately 1 to 3 seminiferous tubules. So we are saying if this is the seminiferous tubule, we are going to take a larger section of it. Okay. So here, the seminiferous tubules, the germinal epithelium, it consists of mainly two types of cells. Do you know the name of these two cells?
Leydig cells and Leydig cells.
It's not Leydig cells. Spermatogonia, yes? Yes. So these cells, these are the spermatogonia, or we say the sperm mother cells, and these elongated cells, they are the Sertoli cells which provide nutrition for the sperms. So in spermatogenesis, first we are taking a sperm mother cell or a spermatogonium. Okay, I am drawing it over here. This is the sperm mother cell or the spermatogonium. The ploidy, as you all know, it is diploid. All the cells in our body are diploid except for the final cell, that is the sperms and the ova. They will be haploid because together they will have to fuse and result into a zygote. So they will be haploid. All the other cells will be diploid. So this is the sperm mother cell, and the sperm mother cell will divide. The process used is mitosis, and then two new cells having the same ploidy is formed. These cells, again they will divide by mitosis. Right now I will show only a single cell. These are the sperm mother cells which divide in order to produce more sperm mother cells, or the spermatogonia. This division is meiosis 1. Meiosis 1 is a reductional division. So how many chromosomes do you expect to be in this cell? It is a reductional division. N. That's right. So here also N. Here also N. So this is the primary spermatocyte. Now in the next step of division, this forms the secondary spermatocyte, which will be having n number of chromosomes. So these chromosomes again they develop and form the spermatids. Okay. So basically spermatogenesis consists of three main steps. That is the spermatocytogenesis, spermiogenesis and spermiation. So the spermatocytogenesis is the division till meiosis 1 and meiosis 2 takes place. That is still primary spermatocyte and the secondary spermatocyte. That is, this is the primary spermatocyte. This is the secondary spermatocyte. Here it is meiosis 1 and here it is meiosis 2, the process which takes place. So meiosis 2 is equational division. Equational division means the same number of chromosomes which is present in the primary spermatocyte, they will be there in the secondary spermatocyte. So when you are telling spermatogenesis, the definition is not like the formation of sperms. Formation of sperms, it happens here in spermiogenesis also. From the spermatids, when they are forming a tail, they become the sperms, and that process is a spermiogenesis. So spermatogenesis, when you are telling, you should tell that it is the formation of sperms from the sperm mother cells. So this is the actual process. I'll explain once again. This is the spermatogonia, which is having ploidy 2n. By the process of mitosis, they make new copies. So these are also more number of spermatogonia cells. By the process of mitosis they have done, so they have the same ploidy. Now after mitosis, they move on to become the primary spermatocytes. Again they follow mitosis, so they have two cells will be formed from one cell having the same ploidy. Then from primary spermatocytes, the first process is meiosis 1, which is reductional division. So only half the number of chromosomes will be there in the next cell. So half, in the sense the secondary spermatocytes will be haploid. Then meiosis 2, which is equational division. So again the cells formed which will be having the same ploidy, so they will be n. These are the spermatids. The only difference, these spermatids are circular, haploid and non-motile, whereas these sperms, they are motile. So these spermatids, they do the process of maturation in order to form the spermiogenesis. Like I'll tell the process again. Here it is. Here the process is multiplication. Here it is the growth phase. Here it is the development phase. That is the maturation. And here it is the differentiation. So multiplication in the sense sperm mother cells are multiplying in order to form more cells. Then growth phase is when these cells become large and they form the primary spermatocytes. Then after that maturation is when the meiosis 1 and the meiosis 2 steps takes place. Then after that differentiation happens when the spermatids change into the sperm cells. This is a portion of the seminiferous tubule. Again, it is a recap. Primary spermatocyte having 2 secondary spermatocytes it will form. The ploidy is haploid. Then this one of the secondary spermatocytes will form 2 spermatids having ploidy N. Then finally a spermatocyte, a primary spermatocyte leads to the formation of 4 spermatids or 4 sperms. Now spermatocytogenesis is still here till the formation of spermatid. Next is spermiogenesis. Spermiogenesis is the formation of the sperm from the spermatid, that is the tail part will be formed. The spermatids are non-motile, haploid and circular as I told before. After that the tail will be formed, that is the process which is known as spermiogenesis, and finally like all these processes it takes place over here. So here it is becoming the primary spermatocyte, then the secondary spermatocyte, then is becoming the spermatids. Finally, it will obtain the nutrition from the Sertoli cell. It will put its head inside the Sertoli cell and it will take all the nutrition, food, everything from it. And this is the lumen. Lumen actually means the hole, which is like in our blood vessels, it is hollow, right? So the blood will pass. Like that, everything has lumen. So this is also having a lumen. So these spermatids which are matured, they are released through this lumen, and that process is known as spermiation. So the mature sperms come out of the body by the process known as spermiation. And then why do you think all these process, like it does not happen in males when they are small, it happens at puberty? Why do you think all this happens at puberty? Hormones, yes. So all these things happen at puberty due to the secretion of some specific hormones. So when the boy child is attaining puberty, the hypothalamus secretes GnRH, which is known as the gonadotropin releasing hormone. So I'll write that. Hypothalamus releases GnRH, which is the gonadotropin releasing hormone. This acts on the anterior pituitary, anterior part of the pituitary. So as it acts on the anterior part of the pituitary, two gonadotropins are released. These are the FSH, that is the follicle stimulating hormone, and the LH. This follicle stimulating hormone, it gets mixed with the blood and it enters into the Sertoli cells. These Sertoli cells are triggered due to the release of the follicle stimulating hormone. And the luteinizing hormone, that is LH, it triggers the Leydig cells. Leydig cells are the interstitial cells which are present around the, I drew a structure here before, that is the cross section of the seminiferous tubule. Outside that, these Leydig cells are present. These are known as the interstitial cells. So these Leydig cells, when they get triggered, they produce some androgens, and these androgens are the raw material for testosterone. So they turn on to become the testosterone, which is causing the secondary sexual characteristics in the male. And here the Sertoli cells are also triggered by the FSH, and it releases some factors. Actually only this much is there in NCERT, but then Sertoli cells, they are actually releasing androgen binding proteins. When these androgen binding proteins are secreted by the Sertoli cells, some of the androgens will form the testosterone, whereas the rest of the androgens, testosterone formed will bind down with the androgen binding proteins, and as a result the spermatids become the sperms. So this is how it affects the process of spermatogenesis. So the human male ejaculates about 200 to 300 million sperms during a coitus, and for normal fertility, at least 60% of the sperms must have normal shape and size, and 40% of them must show vigorous motility. This is a video. So I'll explain this process of spermatogenesis. In the testes, in the testicular lobules, in the seminiferous tubules, you can see that this is the epithelial cells, and here you have the sperm mother cells as well as the Sertoli cells. The sperm mother cells are undergoing division, mitosis, and they make more copies of themselves. Then again, by the process of mitosis, they are going to make primary spermatocytes, which will be diploid, which will be having ploidy 2n. After these primary spermatocytes are formed, then by the process of meiosis 1, the secondary spermatocytes are formed. Meiosis 1 is a type of reduction division, so it will be having only n number of chromosomes. And again, meiosis 2 takes place in order for the formation of spermatids, that is equational division. So all these four will be having only n number of chromosomes, that is, it would be haploid. So these spermatids, the cytoplasm, they'll shed, and the acrosome is formed, then the tail is formed, and as a result the spermatids change to become the sperms by the process of spermiogenesis. And after these cells are matured, they obtain nutrition from the Sertoli cells, and after that they remove their head from the Sertoli cells and they move on into the lumen, and they come out of the body, of the male body. So yeah, that's it.
After the formation of sperm, are they stored somewhere before ejaculation takes place?
So the question was, after the formation of sperms, are they stored somewhere before ejaculation? Yes, they are stored in the epididymis cells before ejaculation. Yes.
In the process of spermatogenesis, could you tell me which gonadotropin induces the process of spermatogenesis?
Here two gonadotropins are released, that is the FSH and the LH. Here the FSH, that is the follicle stimulating hormone, induces the process of spermatogenesis through this process.
Is there anything in particular that stimulates the hypothalamus to secrete the GnRH?
When it is like the person attains puberty, the hypothalamus receives a signal and it secretes the gonadotropin releasing hormones, which triggers the anterior part of the pituitary gland, that is, it has a gonadotropic cell which is stimulated and it releases two gonadotropins, FSH and LH.